Hi, good morning. I hope all of you enjoyed your long weekend. Um, Today we're going to shift gears a little bit and start talking about how transforming cells and cancer cells interact with their surrounding tissue environment. In other words, the other cells that are right there with them. Uh, what are the types of interactions that take place, and why is appreciating the interaction between transforming cells and the supporting cells around them so important and relevant to our understanding of how cancer progresses, especially in terms of the spread of cancer, metastases. So this is what we're talking about when we say heterotypic cell interactions. And the role that stromal cells play in them. Okay. So some key concepts. Up until now, in the semester, we've been very sort of content, just kind of thinking about different pathways inside of a cancer cell in a kind of more reductionist way, right? Because oftentimes when we study different pathways, and you know this from looking up different papers about different pathways, they tend to talk about a particular pathway in isolation. Right? And you know, the benefit of that is it's easier to understand how different proteins might play different roles in a particular pathway. And it's easier to understand maybe how a mutation in this particular gene that makes this particular protein, because it fits in this pathway, could have downstream impact. But of course, right, one thing that I think all of you appreciate by now is that when we're talking about intracellular pathways, there's so much overlap. Remember, some of those slides that I've showed you, right, are really complicated. All these different network of pathways that are influencing each other, crisscrossing, potentially interfering with each other. So if you take this on a larger scale, not just the pathways in the cell, but the overall phenotype of the cell itself, right? In particular, a cell that's maybe like transforming or is becoming cancer, right? The cell itself doesn't exist in isolation. You'll recall that normal cells, for example, epithelial cells, because that's what we discuss mostly in this class, get signals from neighboring cells and tissues that are telling them what to do, right, and how to behave. So here's a question. Do the communications still exist between the cancer cell and its microenvironment around it? So up until now, we've been mainly talking about cancer as though it's just the cancer cell doing its own thing, almost like a monologue, right? If we were to use an example of, for example, like a play or something like that, right? A monologue, of course, is one character who goes off on this extended, speech or whatever, right? But what we really want to appreciate now is that in reality, a cancer cell exists as part of a dialogue, a discussion going back and forth between the cell and the surrounding tissues. We already know, right, in this class that things like epithelial cells are heavily dependent upon the messages that they get from other tissues. So the epithelial cell, we've seen this slide before. If you have epithelial cells, they sit on a basement membrane that keeps them anchored, right? That lets them know that they're in the right place, and so that they should feel free to continue doing their normal differentiated cell functions. Right? When they detach from the basement membrane, that's when the epithelial cells are often told to undergo apoptosis through that funny anoikis pathway, because, right, we cannot afford to have epithelial cells just falling off of different linings within our body, floating around, and then you end up with like a lung epithelial cell somehow lodged in your brain and continuing its function as a lung cell in your brain, right? You don't really think about that, but yeah, if you think about it, that's kind of obvious that we had to have evolved so that you don't have the different cells in your body just spreading around all over the place and still being happy and continuing to grow in their normal kind of way, right? So that is a very good example of how epithelial cells depend upon messages that are coming through the basement membrane from the supporting tissue underneath. And that supporting tissue underneath, right, is what we call the stroma. So in this image here, you can see here's the basement membrane, and beneath are stromal cells and a network of different types of proteins that make up the stromal environment. Here is a similar image, but done with immunohistochemistry staining. So in orange or red, they've stained the proteins of the basement membrane. They've stained the nuclei of the epithelial cells. So you can see in blue, right, they're all kind of resting on the basement membrane. And there are important proteins that help tell the epithelial cells that they are successfully communicating with the basement membrane and also each other, right? So for example, adhesion molecules like E-cadherin proteins that are stained in green. So you see that in between the epithelial cells, letting them know that they are in the right place, nestled up against a neighbor. So that's all fine and good if these epithelial cells are normally behaved, right? But what happens during cancer? What happens to this communication? So let's start off by first taking a closer look at the importance of the basement membrane. So the basement membrane, as shown in this cartoon here, is really like just a thin region of the extracellular matrix. So if we look at this illustration, the gray surfaces represent the cell membrane of the cell. So you have this phospholipid bilayer, right? So you have a phospholipid bilayer here, the plasma membrane. And of course, within that membrane, you have all kinds of proteins that are kind of anchored there. Many proteins have to do with sensing the environment right around it. So the environment right around the outside of the cell would be the extracellular matrix of proteins. In this case, you can see that extracellular matrix is made up of different types of fibrous proteins like laminin, collagens, for example, that form kind of this mesh, right, of proteins. I mean, it looks like basically you've thrown like a fishing net over it, right? And importantly, you can think about it that way. That's what it does. Not only does it provide an anchor point, because in the membrane, you can see these green things are these various integrins. Remember, integrins work as a dimer of an alpha and a beta integrin molecule. And depending upon the shape that they create by being paired up in different ways, they can attach to different proteins that are in the extracellular matrix. You can see these are bound to the blue laminin ones. I'm sure you'll have some examples in here of other examples of proteins attached to the collagen fibers that are going back and forth. So not only does this extracellular matrix provide an anchor point, but also, because it's like a mesh, it represents a place where different signaling molecules, right, might actually be held or prevented from reaching the surface of the cell. So many signaling molecules like growth factors and things like that, remember we've learned they have to be received by epithelial growth factor receptors that are on the surface of the cell, like for example, different types of receptor tyrosine kinases. Imagine now, right, that this signaling protein like this growth factor, it's just not floating in water up to the receptor. It actually has to make its way through this mesh-like extracellular matrix. And many times, right, depending upon how thick this extracellular matrix is, you can really limit how many of these signals actually make it to the surface of the cell. So this network, this ECM, can be filled with different types of signaling molecules that are, just a little bit kept away from binding to the surface proteins like receptors on the of the target cell, right? So with epithelial cells in particular, this extracellular matrix we call the basement membrane, right? Because this is a particularly dense region of extracellular matrix. So keep that in mind, right? Extracellular matrix provides anchorage for things like epithelial cells, and it provides sort of a reservoir of different types of growth factor signals, other types of cell signaling molecules that just get stuck and held in the ECM, right? And then are not able to reach the surface of the cell. Let's look at some examples of how the stroma right, beneath the basement membrane can actually vary quite a bit depending upon the type of cancer that you're looking at. In this case, we're looking at two slides of two different subtypes of breast cancer, breast carcinoma. Okay, so in this case, on the left is what we have ductal carcinoma, and on the right is lobular carcinoma. So both of these are carcinomas, right? So you know that these are both involving epithelial cells that started off as healthy breast cells. In the left here, the epithelial cancer cells are stained in brown, and the surrounding tumor stroma cells, right, that are all part of this tumor, this is a big cross-section, are stained in blue. On the right, the epithelial cells, the cancer cells, are stained in blue, and the tumor stroma cells are more this black color. Look how different, right, not only the epithelial cancer cells are, because that's not surprising, right? Because after all, cancer arises from random mutations. So that's why even though you're talking about, oh, this type of cancer is breast cancer, right? Even within that category of breast cancer, you would have different types of phenotypes resulting from different sort of mistakes, different sets of mutations that create one type of breast cancer cell that phenotypically is very different than another breast cancer cell. So look at the difference not only in the cancer cells themselves, but the impact that they have on the stroma. Right? So very clearly, the stromal tissue here is heavily influenced by the cancer being present. Right? And so this is what we're talking about when we say there's dialogue going on. Right? So what is stroma? So far, we've just treated stroma as just being like, oh, it's the cells that are beneath the basement membrane of a layer of epithelial cells, and these stromal cells send things like mitogenic, growth factor signals, or maybe signals that favor differentiation, right? And those signals have to get through the basement membrane and reach the epithelial cells. But let's look at this in a little bit more detail. So the stroma is a complex tissue made up of many different cell types. So here listed in brown, red, and blue are just examples of maybe like 9 groups of different types of cells that make up stromal tissue. These can be thought of as providing, one of 3 different type of important stromal functions. For example, structural support. Right? Obviously, you need to have the tissue have some structure so that you could have then the basement membrane and the epithelial cells on top of it. Some of these provide or are part of the vasculature, the blood supply that has to be flowing through here to be able to nourish the epithelial cells above, and not only nourish it by providing nutrients, but also managing gas exchange, right? O2, carbon dioxide. And an important part of the stromal environment are immune cells, things like macrophages or lymphocytes, like T cells, for example, are part of this as well. So if you have all these different cell types coexisting in the same sort of tissue environment, you have to imagine there's a lot of complicated cell-to-cell communication going on, right, in order to maintain this kind of happy, normal, balanced environment. So these communications that are happening between different types of cells, we just in general call this heterotypic signaling. Each one of these cell types is sending messages to either encourage or limit the proliferation of other cell types around it. Right? And it's that careful balance, that fine-tuned balance of these heterotypic signals that keep the stroma healthy and happy so that it can support the epithelium above it. So we tend to focus in this class on carcinomas as a model for understanding epithelial and stromal interactions, right? Because as I've said repeatedly through the semester, we focus on carcinomas and this idea of the epithelium model of epithelial cells sitting on top of a basement membrane, sitting on top of stroma to study cancer. Because it represents many of the important concepts of how cancer happens and, importantly, how cancer metastasizes. But I always want you to keep in mind, right, there's many types of cancer that have their own separate sort of model for understanding these types of interactions. Right. Some of the earliest experiments done in the 1950s or so to help shed light on the importance of stroma somehow supporting these cancer cells, right? So the important thing is, I think you may have wondered about this. Certainly early oncologists wondered about this. Once a cell becomes a cancer cell, does it really care about the stromal environment that it's in, right? Or is it so messed up and independent that it can just do whatever it wants and it doesn't matter, right? So as it turns out, and I don't think you'll be surprised about this, right? Even aggressive cancer cells, epithelial cancer cells like carcinomas, still depend very heavily on the environment of stromal cells to be able to continue to support them. Right? So tumor cells expect stroma to support their growth. So back in the 50s, before we had a lot more, moral, ethical kind of awareness of how to do cancer research, and certainly well before we had patient rights and the protection of patients. You remember the story about how HeLa cells come from cervical cancer epithelial cells taken from a woman who never consented to have this done, right? And she died just a few months after her cervical cancer cells were taken by her physician without her knowing, and introduced into a cell line or grown into a cell line in the lab, right? And so to this day, her cancer cells live on as HeLa cells. These days, you cannot do that, right? In the early 50s, there were even worse examples of this. So this cartoon actually represents the type of experiments that were done, where tumor cells were taken from their original location. For example, if this individual had a tumor in his arm, right? And were actually, transplanted to a different part of the body, like the back, to see what would happen, right? And you're like, what? Hopefully you're appalled by that, right? I have no idea how they got away with this kind of stuff back then and what they told the patient they were doing, right? But what they found, okay, so you give them some credit because it's because they were able to do kind of barbaric stuff like this that they made some important discoveries, right? And that is when the epithelial tumor cells are transplanted alone, just the actual cancer cells, from the arm to the back, the cells struggle to grow, right? So they struggle to form a new tumor on the back. However, when some of the stromal cells surrounding the tumor cells were also transplanted with the carcinoma cells, then they saw that tumors would start growing on the patient's back. So here you see, if you transplanted the carcinoma cells with some stroma, you can see they begin to grow and they look like they're actually forming, right, a new sort of like tumor and a new site here, right? Without the stroma, the cancer cells, they're just lost, right? They're really confused. They don't know how to organize, and you don't get a tumor formation. Okay. All right. So that means metastases, in other words, the migration of cancer cells from the original site of the original primary tumor spreading to other tissues in the body, right? That's metastases, is dependent on those cancer cells still having a dialogue with stromal cells wherever they end up, right? Not only wherever they end up, but even as part of how they spread in the first place, how they leave the primary tumor. So different types of tumor epithelium, right, when they spread to different locations in the body, they modify the surrounding stroma to sort of suit or fit their needs. It's not surprising that cancer cells that are very rebellious, right? They're very independent, so they cannot function by themselves. But what they can do is they can trick or force the stroma surrounding them in a new location in the body to kind of tend to, their needs. Right? A good example of this is, look at this here. So here we have two examples of liver tissue so this is normal liver tissue, where metastatic cancer cells have arrived in the tissue, right, and have now set up the formation of a metastatic lesion or a new metastatic tumor. On the left, those cancer cells happen to be colon cancer cells, right, that came from the colon and have spread to the liver. And on the right are breast cancer cells that have made their way to the liver, through the circulation, for example. Right? And what we're appreciating here is how different, right, the end result can be. Keep in mind that both of these are taking place in the liver. The colon cancer cells, which are the sort of dark blue ones here, right, they begin to communicate with the liver stroma, and they alter the liver stroma. On the very left would be the normal liver cells over here, right? And then in this region in between is the stromal cells that are beginning to be changed by the presence of the tumor epithelium growing over here. And you can really see how the tumor, the stroma, looks quite a bit different than the normal liver, right? And what's surprising is because the colon epithelial cells, they came from their original environment where they were, parts of things like ducts or different types of crypts in the wall of the colon, right? That's what these epithelial cells, that's what they grew up sort of expecting, right? An environment where they're surrounded by crypts and ducts and things like that. And so that's what they trick the liver cells into providing them, right? They trick the liver cells into providing them different types of support so that the tumor epithelium begins to form things like ducts in the liver, right? In this type of tumor. That's quite different than what the breast cancer cells do. Here you have on the right the normal liver cells over here, and then here you have the metastatic breast cancer cells moving in. What you can sense here is, in this case, the breast cancer cells are really shoving the normal liver cells aside, right? Trying to get access to the vasculature. So throughout this normal liver tissue here, you can see little red spots, right? These little red spots are red blood cells. That means anytime you see a little group of red spots here, that happens to be a cross-section of a capillary that was running through here, right? And so these breast cancer cells are shoving and telling the normal liver cells to spread apart so that the cancer can have access to more of the blood flow, right? So again, in both of these cases, the colon cancer or the breast cancer arriving in the liver, there's going to be dialogue taking place between the cancer cells and the new stromal environment, right? But depending upon what the cancer needs, right, phenotypic changes in the stroma take place can be very different to support the growth of the new tumor. So I think about it this way, right? I think about like a transforming cancer cell as a kid who's now become a teenager, right? And, a typical teenager begins to feel sort of rebellious and wanting to be very independent of his or her parents, right? So, in this case, so she, she's like rolling her eyes, right? My daughter is 10 now, and I swear she's practicing to be a teenager, because now anytime I ask her to do anything, she like rolls her eyes, right? And she like sighs, right? So I can imagine, right? So a teenager, right? She's in her room, probably online with her friends complaining about, oh, my life sucks. My life is so hard. I hate life. My parents don't understand me, blah, blah, blah. They wouldn't care if I died, I should just run away, blah, blah, blah. Right. But then the moment her mom calls from downstairs, and her mom's like, honey, dinner's ready. I made your favorite lasagna. Right. And this girl's like, okay, I got to go. Right. And then she's downstairs. So the point is, right, tumor cells are beginning to rebel. Right? Remember, they're beginning to lose their dependence on properly listening to the types of signals that the surrounding stroma are providing them. Right? Because the surrounding stroma might be saying, hey, no, stop proliferating, right? You need to just focus on differentiating. But the transforming cell, right, because of oncogenic mutations, loss of tumor suppressor genes, right, is beginning to say like, no, forget that. I'm doing my own thing, right? But the important thing to keep in mind, right, is just like this teenager here, right, even though she's rebelling and she thinks of herself as being really independent, she's still living in her family's house. That means she still depends upon her parents for support, right? And even though her parents get really confused and they start getting manipulated by their teenage daughter, right, the important thing is they still provide this type of environment where their daughter can still, thrive, right? I know many of you are like, I don't understand this example because I was never like that as a teenager. Yeah, right. Okay. I was like this as a teenager as well, right? But that's another story. Okay, so I want you to think about that, right? The transforming cancer cell as the rebelling teenager who begins to no longer listen to parental advice or the stroma, right? But at the same time is very manipulative, right? And is causing the stroma to get very confused and can kind of manipulate the stroma into kind of helping or supporting their own sort of misguided notion of what they want to do. But importantly, this is still happening within the original tumor environment, the primary tumor site, right? We're not even talking about metastases yet, right? We're just talking about this dialogue that's beginning to kind of break down between the cancer cell and the stroma. Not break down meaning it's being lost, but it's becoming very dysfunctional. Right? So how do the tumor cells do this? Do tumor epithelial cells, do they come up with novel ways of manipulating the stroma, or do they actually make use of existing pathways, right, that the stroma is already sort of geared up to perform? I think you should probably already know where this is going, right? Because all the way since the beginning of the semester, remember that first example of the orchestra, and I used Eddie Van Halen, the rock guitarist, right? And I asked, is a transforming oncogenic mutation, a transforming cell, like Eddie Van Halen, crashing an orchestra and all the musicians freak out and fall over, knock over all their little folding chairs and stuff like that, right? And we said, no, right? Mutations that alter intracellular signaling pathways, right, are alterations in existing pathways that become either amplified, like oncogenic, right, or lost, like loss of tumor suppressive pathways. So that same theme applies to how transformed cancer cells communicate with their stroma surrounding them. Right? In other words, just as transformation of the cell itself involved deregulation of existing intracellular pathways, so do the events that allow the tumor cell to alter its surrounding extracellular microenvironment. In other words, tumor cells must somehow trick the surrounding tissue to help them thrive, expand, and eventually invade, right, the surrounding tissue. And the most effective or efficient way for the tumor cell to carry out and get the stroma to do its bidding is to activate a set of normal physiological responses that these stroma cells, as part of the normal healthy tissue of your body, are already prepared to execute, right? I hope that makes sense, right? Because when the time comes and the tumor is growing, right, and it's beginning to really expand and really begin to grow really large, it needs to have the support of the stroma, and that convincing of the stroma to help it out, help the tumor out, is not going to have the luxury of taking 5, 10, 15 years more of random mutations, right, to work. Right? When the time comes, this actually happens pretty quickly because the tumors, through their random mutations, somehow learn very quickly to activate a set of pathways in the stroma that end up supporting. So what set of pathways can you think of, right? What are some things that maybe normally are kept kind of quiet in our tissues, but if they're needed, they need to actually activate pretty quickly, right? And might represent a pretty complicated set of different changes taking place in the tissue. Right. Well, since, normally if we were together in the classroom, I'd sort of expect you guys to kind of like chime in here. But I'll just tell you, right, for the sake of time also, tumor formation has many parallels with wound healing, right? Because wound healing is what I'm talking about. Let's imagine, right, you have your arm and the tissue in your arm is perfectly normal and happy, doing its normal functions, right? It's minding its own business, everything's good. But if you actually get injured, like you get a cut in your arm and there is damage to the tissues, right? Yeah, you can imagine that a whole set of pathways turn on very quickly, pathways that we evolved to be able to activate right away, so that changes could happen very quickly to do things like stop the bleeding and begin the healing process, right? So this is a flow chart of different wound healing responses. So keep in mind, what you see here has to do with normal, healthy tissue that, for example, your dermis in your arm, right, getting injured, like with a cut. And we'll divide this into the stroma cells that are supporting the epithelial cells. And we'll take a look at the changes that are happening in each one of these pretty quickly. Right? So in the stromal cells, let's start off here, the top. Right? So there's damage to the stroma, right? So you got a cut and some of your stroma is now injured. There's going to be damage to vasculature, right? The capillaries that were in that area, they're going to be torn. You're going to have blood leaking out, right? I mean, you're bleeding, right? So what happens? You're going to get the damage to the vasculature signaling different things, like the aggregation of platelets, right? So the platelets in your blood will begin a blood clot to try to stem the bleeding. Okay? There will be exposure of plasma, plasma that's leaking out of your blood, right, into the surrounding parenchyma or stroma. And so that will trigger conversion of a lot of fibrinogens over to fibrins that will then serve with the clot. Platelets that are arriving, when they're at the site of the damage, they will degranulate, meaning platelets will release different types of signaling molecules. Things that are, for example, vasculature factors that encourage blood supply, right? So in this case, the blood will be encouraged to have more plasma leaking out. Importantly, it will also release these types of signaling molecules like platelet-derived growth factor and TGF-beta. So when you look at a normal capillary network in healthy normal tissue, this is a neat image, right? Because obviously the capillaries are stained in red. You can see they're very organized, very neat, and very tidy, right? All the blood is contained within the capillaries that are flowing through this capillary bed. So that would be in a normal healthy tissue. Let's look at what the capillaries look like inside of a tumor. That's what you see on the right. Ooh, what do you notice? They're really leaky, right? They're much larger. They're very leaky, so a lot of blood leaks out. What does that mean? So that means the tumor blood vessels are already leaky. That means they already appear as though there's an injury there, right? So there's plasma seeping out into the surrounding tissues from the tumor. Platelets are being recruited there. The tumor cells themselves may have mutations where they've learned to secrete things like the PDGF and the TGF-beta, right? That normally the platelets are supposed to take care of. But remember, PDGF, TGF-beta, these are proteins, signaling molecules, so they're coded for by genes, right? So all of the cells in your body that have a genome in it have these genes. It's just normally, right? These are not genes that any old random cell would bother making, but platelets, contain these proteins that are made by the megakaryocytes before they break up as platelets and are carrying these proteins around. So random mutations in the cancer might activate things like PDGF and TGF-beta production as well. Okay. So the next step then would be the recruitment of certain types of immune cells. So macrophages will come, again, because it sort of looks like there's an injury here, right? So macrophages will come as part of their normal function to kind of help try to fight any kind of infection that your body expects might potentially take place if you have an injury. Right? So you get the formation of recruitment of macrophages, and also the recruitment of different fibroblasts to lay down, right, different new connective tissue there. These generate a lot of enzymes like matrix metalloproteinases, so MMPs. Right? So as the name implies, these are enzymes that break down proteins, proteinases, and they utilize different metal ions in order to have the right shape to function. Why do macrophages do that? Normally, if you have an injury, right, the tissue that's still there, right, might still be like really damaged and not usable tissue anymore, right? So part of healing is actually the further breakdown and recycling of damaged tissues, right? For example, in your skin. So the secretion of these matrix metalloproteinases helps actually break down the stroma in order to make it easier to kind of recycle and replace with new tissue. And also the breakdown of the stroma encourages more of these types of blood factors to be able to get into the site of the injury. As part of that, they also produce vascular endothelial growth factors that encourage the growth of new capillaries to replace the ones that were damaged. So here's a table looking at some examples of metalloproteinases. You don't have to memorize this, but I do want you to look at it and realize that these different matrix metalloproteinases, these MMPs like 1, 2, 3, etc., right? Their alternative name oftentimes makes more intuitive sense, like collagenase, gelatinase, etc., right? Because they have to do with digesting the damaged stroma at the site of an injury to be able to remodel the tissue, clean it up to make way for new tissue to grow. Right. Here are some properties of vascular endothelial growth factors. Right. So these signaling molecules that are released encourage capillary and lymph duct formation. They encourage monocytes in the blood to migrate to the area and become more macrophages to help maybe fight potential infection. They have hematopoietic properties because you're going to have to now replenish or regenerate more different types of differentiated cells in the area. Regulation of endothelial cell pools. So as the blood vessels are forming and you have the endothelial cells lining the blood vessels, right, these VEGF signals help manage that. And they also help maintain the integrity or the permeability of the capillaries, both the existing capillaries and the newly forming ones. All right, so that brings us to the bottom of this. So the matrix metalloproteinases end up digesting the extracellular matrix, right, to get rid of the damaged parts of tissue to be replaced. What does that happen? I mean, what happens when the ECM gets digested away? Remember, the basement membrane is an example of a type of ECM, and we said the basement membrane is important for what? Forming a physical anchor point for epithelial cells. That was number one. And number two, it represents a reservoir of different types of growth factors and different types of mitogenic signals, right, that never quite reached these cells because they were caught in the basement membrane. But now if you're digesting away the proteins of the basement membrane, you get the liberation of all of these growth factors like cytokines, GFs, things like that. Right, that suddenly now are released, right, and can signal and reach the surface of many of the other cells in the area. Right? Normally, that's to encourage the growth of healthy tissues. Also, you'll get the formation of new blood vessels. So this is a process known as angiogenesis, the growth of new blood vessels. We will look at angiogenesis later. But not surprisingly, that's encouraged by vascular endothelial growth factors, right? And you get the growth of new capillaries into your injured tissue. And ultimately, you have epithelial cell proliferation taking place, right? So we can then shift. All of that is happening in the stroma in response to a wound, right? What happens to the epithelial cells? So the epithelial cells that border the site of your injury, right? So let's imagine you have, a nasty cut, right? And so the epithelial cells on either side of the cut, those are the ones that you want to now proliferate, right? Proliferate and migrate to be able to fill in the area of the cut where you lost your old epithelial cells, right? But remember, epithelial cells normally want to stay put on the basement membrane, right? In order to encourage these epithelial cells to not only let go of the basement membrane and migrate and sort of give up their differentiated job temporarily and become more focused on proliferation, right? They have to undergo a change. And this is the EMT. We've heard about this throughout the semester. We're going to talk a bit more about it. But I want to introduce that in greater detail now because hopefully you understand. Epithelial to mesenchymal transition, right? The changing of epithelial cells from an epithelial phenotype to be more of a mesenchymal phenotype, right? Mesenchymal meaning the stromal phenotype that allows them to be able to be happy, detached from a basement membrane and allows them to want to proliferate more rather than being just fully differentiated cells. So that's EMT. So these epithelial cells will move into the wound site, they will proliferate and cover the wound, and then importantly what? They have to then, once the wound is filled in, revert back to their normal epithelial status. So the reverse of EMT would be MET, mesenchymal to epithelial transition, right? And once that happens, you've now reconstructed the damaged tissue. And maybe if you're lucky, all you have is a tiny little scar there, right? So I want you to think about this, okay? Everything we've talked about on these flowcharts, this is what normally happens when you have a wound, right? All of these pathways are ready to go at a moment's notice because we evolved that way, right? You cannot wait hours and hours or days for these different parts of these pathways to suddenly get upregulated and, oh, I need to make more protein for them. Because meanwhile, what's happening, right? You're bleeding out, right? And bacteria is getting in and everything, right? So our cells have these pathways ready to go. And all it takes is certain triggers to activate them. And a series of events happen pretty quickly, right? So the reason I say this is I want you to consider this. Tumors behave like internal wounds that are always healing, right? Whether it's the leakiness of the blood vessels or mutations that allow the tumor cells to produce signaling molecules like PDGF, like TGF-beta, like VEGF, right, vascular endothelial growth factors, right, or even things like matrix metalloproteinases, right? This is the scary thing. All of those products are coded for by genes in your genome, right? And so all of those genes potentially can get mutated and turn on improperly in the formation of a tumor. So if the tumor learns the right combination of these things through mutations, it appears to the surrounding tissue, right, that it is just a wound that needs to heal. And as a result, the surrounding stroma activates these wound healing pathways that are shown on these flowcharts. And unfortunately, whereas if it's really a cut in your arm, these are the pathways that you want to happen quickly to result in migration of epithelial cells, proliferation of epithelial cells, right, in order to heal. But if these are the things that a tumor is tricking the surrounding stroma to do, ironically, these types of things, right, encouraging epithelial cells to be okay, letting go of basement membrane, encouraging epithelial cells to be more mesenchymal so they can move around, right, encouraging more blood supply to feed the tumor, all of these things, right, are dramatically detrimental, right, to the body because these are now suddenly going to encourage a new phase of the tumor's growth. Right? That's why I want you to keep in mind, we said that it takes many, many years of random mutations from that original starting transforming cell to reach a large tumor mass, right? But oftentimes, it's only at that point when that mass is detected, because that's when the patient, especially if the tumor is very deep in the body, that's only when the patient begins to feel symptoms, pain, discomfort, right? Maybe there's some blood, in the stool or something like that, right? But by then, the tumor is very large, and it seems like you hear about somebody who was just diagnosed with cancer and they're in the hospital, and things go downhill very quickly from there, right? Because once that tumor reaches that size where suddenly there's selective pressure for it to activate these types of wound healing responses in the surrounding stroma, if it succeeds in doing so, right, we are a victim of the efficiency of our own wound healing response, because in that situation, then the tumor will get all of this support that it was really needing to suddenly go really out of control very quickly. Okay. This is the last slide for today, because I wanted to talk to you about the connection between our immune system and things like developing tumors, right? So a big part of our surrounding stroma around something like a tumor are actually immune cells, right? So immune cells are a big component of our typical stromal tissue. And the immune cells themselves can be fooled into helping the tumor. So in this case over here, you can see here are carcinoma cells in blue, and the surrounding tissue is stained in brown, because what they're staining for is the matrix metalloproteinase 9 enzyme that is being secreted by these macrophages, these immune cells that have been attracted by the tumor through different types of wound healing signals that the tumor is giving off, right? And these immune cells are being fooled into thinking, oh, this is a wound that we need to now remodel the stroma. And so these TAMs, or tumor-associated macrophages, are secreting a lot of these matrix metalloproteinase enzymes that are basically now remodeling the surrounding stroma, ironically making it much more accommodating for the carcinoma cells to now really start expanding and growing, right? So I think this is a really good example of what we're talking about when we say the cancer cells don't operate independently, right? They trick or fool or usurp the functions of surrounding heterotypic cells in their normal stromal environment to help them do their bidding. Right? Okay, let's go ahead and stop there today.